Okay, now we'll be discussing 8th VSAQ. The question here comes is, 8th VSAQ is, what is downstreaming process? What is downstreaming process? Or how can you define this process, we can say, and what is it useful for rather? Okay, downstreaming process. It is a process of separation. It is a process of separation and purification. So separation and purification of what? So as we are telling, that downstreaming process is the separation, that is making the part, then purification, taking the purified of what? Of desirable clones, or not only the desirable clones, or the gene products. Desirable clones or the gene products which are obtained from this process. Okay, so it is a process of separation, purification of desirable clones or gene products. Actually, to say this is downstreaming process is used at the end of or in order to separate these desirable clones or gene products we are telling. This is actually, this process or rather this technique is used in order to separate these products which have been attained as a result of genetic engineering technology. By means of, or rather by means of recombinant DNA technology, whatever the products which have been obtained as a lump sum, will be getting all the products. So we have to get the actual product or actual clones. So these two products, or rather the desirable clones or the gene products, will be separated through this downstreaming process. And as the question is random, that what is downstreaming, we have just now defined that downstreaming is a process of separation, we are telling, as well as the purification of desirable clones as well as of gene products. When this downstreaming process is done, this downstreaming process is done usually at the end of biosynthetic phase. This is done at the end of biosynthetic phase, and before what? So what you can say simply, downstreaming process is an intermediary process which is going to take place in between biosynthetic phase and the product which has to be, or which has to be also, we can say, which has to be marketed for commercial purpose. Okay, so downstreaming process is a process which is done at the end of the biosynthetic phase and at the beginning or before the product which has been attained has to be marketed. Actually, it has to be introduced to the market, because whatever the things which are going to get from that of the gene products, or rather the clones, desirable clones, are only meant for the commercial usage. For example, insulin. Okay, this is an example also we can take over here. So whatever the products are there, these products has to be marketed. So before they has to be marketed, they has to be furnished in an, or they has to be in an finished level. So they have to attain an, or acquire an finished texture or a furnished texture. Finished or furnished texture, we can say. So downstreaming process is the process of separation, purification of desirable clones and gene products. Desirable clones and gene products. This downstreaming process, it is usually done at the end of biosynthetic phase as well as just before the product is released into the market as an finished, or rather it is as an furnished product. So this is what, and as I have already told you that desirable clones or gene products are usually attained by using genetic engineering technology, that is the recombinant DNA technology, wherein a single gene or a single desirable gene can be multiplied into number of copies. Whatever the gene responsible, okay, or responsible in order to, for example, just now I have mentioned about insulin. Okay, so the gene which is responsible for producing this hormone, that is insulin, and this gene can be isolated, this gene can be multiplied into number of copies, that is called as the desirable clones, and these are also the gene products, these are the products of the gene. Okay, so which are attained by the biosynthetic phase, they have to be released into the market as an furnished product. So this is what a downstreaming process means, the definition as well as when it is done. So here we have end with that of the 8th VSAQs, that is the downstreaming process. Now we'll deal with the 9th VSAQ. Okay, 9th VSAQ is what is green revolution and who is the father of green revolution. Okay, so coming to that of green revolution, so green revolution is increase of or increase in yield of crops. Increase in the yield of crops. Okay, so in order to increase in the yield of the crops, so in order to increase the yield of the crops, what is done? Both the conventional as well as non-conventional, conventional and non-conventional methods, conventional and non-conventional methods of breeding practices, or rather breeding methods you can say. Okay, green revolution is in order to increase the yield of the crops, and the yield of the crops, or what we can say is that green revolution, the target of the green revolution is to increase the yield of the crop. How this yield of the crop can be increased? This yield of the crop can be increased by both the conventional as well as non-conventional methods of plant breeding techniques, as well as we can also use better agricultural practices, better agricultural practices, better agricultural practices. What for all these? These are all implemented only to increase the yield of the crops. Why should we increase the yield of the crops? We should increase the yield of the crops in order to meet in order to meet the human population, or in order to meet the requirements of the human population across the world. So green revolution can be defined as, in order to increase the yield of the crops by utilization of or by using of both conventional as well as non-conventional methods of plant breeding, as well as better agricultural practices. Through these, the yield of the crops can be increased, as well as in order to meet, why this yield has to be increased? This yield has to be increased in order to meet the requirements of the human population across the world. This is called as green revolution. Okay, who is the father of green revolution? Here the question is, the father of the green revolution is Norman E. Borlaug. Norman E. Borlaug is the father of green revolution, and the word green revolution was first used by the scientist named William Gard. The scientist named William Gard. William Gard in the year 1968 has used this term green revolution. Norman E. Borlaug, as he was the person who first introduced this green revolution, and he is regarded as, that is why he is regarded as father of green revolution. And you must be having a question in your mind, then what about M.S. Swaminathan? M.S. Swaminathan is Indian green revolutionist. He is the Indian green revolutionist. M.S. Swaminathan and his students, or rather the workers, have done an extensive work in the southern parts, or in India actually to say, who has brought into green revolution. That's what about green revolution. So green revolution, the main target of green revolution is that in order to increase the yield of the crops by utilizing both the conventional as well as non-conventional breeding methods, as well as by using better agricultural practices. So by using these better agricultural practices as well as both conventional and non-conventional methods of breeding techniques, a better yield can be obtained, or an increase in the yield can be obtained in order to meet the requirements of human population across the world. This is called as green revolution. And the father of green revolution is Norman E. Borlaug, and Indian green revolutionist, or father of Indian green revolution, is M.S. Swaminathan. And William Gard in 1968, he has been given importance because he was the person who used this term, who for the first time used this term green revolution. So that's how we have come to discuss the end of the Okay, now we'll be dealing with the 10th VSAQ. Okay, so the 10th VSAQ is name two genetically modified crops. Genetically modified crops, we can also call them as GM crops. These genetically modified crops, just we can give two examples of these genetically modified crops. That is, one crop we can go with that of BT cotton, and another crop we can go with that of, example, just name any two genetically modified crops, in the sense the examples of two genetically modified crops, BT cotton and BT tomato are the examples. Okay, this BT cotton, coming to this of BT cotton, and what is this BT what I am mentioning over here? This term BT here, it represents, or it gives out, it represents a bacterium called as Bacillus thuringiensis. Bacillus thuringiensis is that BT what it refers. It is a bacterium. And this, what is the significance of introducing this Bacillus thuringiensis into this cotton plant? And we are telling them as genetically modified crops. That means this bacillus, a bacterium called Bacillus, or a bacterium named Bacillus thuringiensis, has the capability, or the spores which are produced, by this bacterium Bacillus thuringiensis, produce toxins. Some toxic substances are nothing but the poisonous substances. These spores produce toxic substances, and these toxic substances are harmful to some insect pests. Not to all the insects. We should be very much particular over here. Okay, so BT cotton, BT the word refers to you, as I have been telling you, that BT the word refers to you as Bacillus thuringiensis, wherein the bacterium Bacillus thuringiensis produces spores, and these spores releases or secretes toxic substances which kill insect pests. That particular gene which is responsible in secreting the toxic substances, if at all this gene of Bacillus thuringiensis is introduced in cotton plant. Again remember, cotton, the name of the cotton is, the botanical name of cotton is Gossypium herbaceum, which belongs to the family Malvaceae. And that's what about BT cotton, which is an genetically modified crop, or you can also call it as transgenic plant. And we'll also be discussing the same in case of the transgenic plants which are made resistant against the pest or the pathogenic control. Okay, now coming to that of the other example, that is BT tomato. BT tomato, and the botanical name of tomato is, Lycopersicon esculentum, belonging to the family Solanaceae, which you have studied during your junior intern, this family. Okay, so in case of BT tomato, what you are telling, the BT tomato or the tomato plant is usually affected or infected by an bacterium or by an bacterial pest. That is, the name of the bacterial pest is the Pseudomonas. Pseudomonas is the bacterial pest which attacks the tomato plant. Bacterial pest which attacks the tomato plant. So, if at all, and the toxic substance which is responsible to kill, okay, the toxic substance from that of Bacillus thuringiensis, sorry, not the toxic substance, the gene of Bacillus thuringiensis which is responsible to produce or to release or rather to secrete a toxin which is responsible to kill the pathogen, that is Pseudomonas. That's how these two plants, these are only the two examples we are discussing over here. We can also discuss a number of examples. As the question is restricted over here, name two transgenic plants, or rather name two genetically modified crops we are telling. So that's what we gave, only simple two examples, that is BT cotton and BT tomato. We can also go with that of papaya plant, like that, so many examples we can go with. So that's what about the examples, or rather the two examples of genetically modified crops, that are BT cotton and BT tomato. So we have finally finished discussing section A, which consists of all the 10 VSAQs, and VSAQs are rather more what you can say is that are more productive, or rather they are more helpful for you in order to score full, that is 60 on 60. So that's what about the end of the VSAQs, that is the section A.